Now, once you have aligned your sequences, thanks to the multiple sequence alignment options or methods that we used, you can have an idea about the conservation of parts of these sequences, and this is based on the fact that the highest conservation is maintained throughout evolution for the most important functions. So the higher the essentiality, I would say, of a feature, the high probability that this would be maintained. So this is, for example, the case for promoters or essential enzymes, etc. Whereas the regulatory regions might be generally evolving a bit more faster, like UTR regions, but we're talking about millions of years of evolution here. So the multiple alignment would help you identify what elements would reject a substitution, because usually substitution occurs in neutral DNA and do not occur if an element is functionally constrained. And remember that terms of functional constraints for an element, because this is something that you might be seeing in the databases. So this kind of evaluations is done through a conservation score that is calculated by different tools and approaches or algorithms also that are highlighted here. So these are some examples of them. So a good one would be the GERP, or that stands for Genomic Evolutionary Rate Profiling, and it measures base conservation. And why would we be able to measure this base conservation, or how? In fact, this is done through the estimation of the mean number of substitution in each aligned genome. And this would help you evaluate the neutral evolution rate. So, because this is, we have an evolutionary relationship between the sequences, then the GERP score and the variance proportion in that sequence would be related. So basically, the higher the score, the less variation there would be in the sequence. PhyloP is another one that measures base conservation from non-coding regions. FastCons has been developed as part of the FAST, which is phylogenetic analysis with space-time models. It uses an HMM model, so hidden Markov model, with states of conservation and non-conservation that are stated as C and N. And this is originally developed for like small or medium-sized sequences. And you also have the SciPhi, which is a site-specific phylogenetic analysis, which not only measures but models the patterns of substitution. And in fact, it takes into account the fact that in the other methods, we only model the decrease in the rate of a mutation and not really the selection that is influencing the pattern of substitution. So it seemed a bit complicated, but in fact you have here, in order to ease the process of comparison for you, you have here a Venn diagram that is showing an example of the three ones that are compared here. And what we notice is that, in fact, it seems that about 60% of the elements are shared between the different methods, whereas others seems to be unique to each one. And in fact, this would suggest that the three methods are more likely complementary to each other, and this shows that each one is highlighting different aspects of molecular evolution. SciPhi being, for example, finding much more different conservation in UTRs compared to untranslated sequences compared to the other ones. So you have here an example output with the FastCons conservation score here and the GERP score here, with for one particular sequence, which is the CFTR locus here. So you have the reference if you want to go and have more details. So for whole genome alignment now we have different kind of methods. I won't be going into the details for the sake of time, of course, but I'm just giving you as an example the MUMMER, ultra-fast alignment of large-scale DNA, which is hosted at this website that you have here. This allows the alignment of entire genome, and what is interesting is that you can use complete or draft versions. The MUM stands for Maximal Unique Matcher, so finding the MUMs would be finding essentially the subsequences that occur only once in both genomes, and that would not be extendable anymore. So if you remember, this is the same process as the extension that we had with the BLAST, and it uses what we call a suffix tree-based approach. So basically using, cutting the sequence in different parts and finding the best way to retrieve the sequence. You also have other tools that are available for publicly available prokaryotic genomes, like the WebAct, which is really, really useful also. And you can do these kind of alignments through databases, and we will be seeing one of these databases, which is Ensemble, when you will have your assignment. So let's talk now about the output. The output of comparative genomics, if you remember well, I was talking since the beginning about structure, evolution, and function. So the output in terms of structure would be at the genome level in terms, for example, of base composition, like the percentage of GC. You would be able to see that. You would be able to know if you have a codon bias, a different nucleotide distribution. In terms also of big genomic events, like structural variance events, big rearrangements, you would be able to see sequence conservation and synteny. And for the genes, for example, it could be the gene order. Now for the function, you could be seeing that at the non-coding sequence level for regulatory functions, and you could be seeing this also at the coding sequence level when you would gain insights into the gene functions, etc. So in terms of genomic evolution, the multiple sequence alignments can allow to infer homology, and we'll be seeing what homology means, and estimates also the evolution distance between sequences and organisms. So this could be done for entire genome comparison or for gene comparisons. So there is a very important notion that was one of the central dogmas back to the early 50s, when Monod said that anything found to be true of E. coli must also be true of elephants. And in fact, this didn't take into account the whole picture of phylogenetic context that we know today. And in fact, this would essentially depend on the expected output if you are comparing closely related species or evolutionarily distant species, sorry. So this is basically what we can highlight here with an easy example. So if you are comparing closely related species, what you would gain as in terms of information would be completely different if you are comparing distant species. So if you are comparing these closely related birds, for example, you would be able to detect changes that are really small and are related to very peculiar characteristics of these genomes. Whereas if you are comparing this genome, for example, to this one, you would mainly be seeing only small common parts of them. So this would be mainly used to highlight the differences. So coming back to our very, very simple example, if you remember it, we were talking about this in the beginning of the session. So if you are comparing closely related species, it would be just as comparing these two dishes, for which the only difference that you have is that red pepper here. Now, if I'm telling you that this first one is a spicy one, and the second one, the first one is a not spicy one, and the second one is spicy, and I told you, imagine what kind of characteristics you would be able to compare with closely related species, then you could easily say then that because the only difference between them is the existence of that red pepper here, then the character spicy is related to that presence of the pepper. Now, if we were comparing the first not spicy to this not spicy, or the spicy one to this not spicy, different dish, we wouldn't have been able to say that the character spicy is due to that presence of the pepper. I hope you see the example here, because here you have so many differences between the dishes that comparing them directly would not allow you to have the whole idea of what is causing that specific character, being the character spicy. Instead, you would be able to say that, let's say that this redness is due to that the common things between them, which, for example, let's say it is the presence of a tomato in a sauce. So here, basically, you would need this kind of comparison in order to highlight small differences that are related to a specific character, whereas here you would be comparing completely different things and highlighting, for example, one common feature. So this is what is done when we are inferring homology from a very complex organism to a simple one. So you have here the example of Ebola virus, for which we use these kind of comparisons. So the people there compared 100 available Ebola virus genomes. So they belong to the Filiviridae group. These are completely different from all other viral genomes. So comparing the Filoviridae to other viral genomes allowed people to confidently assign them to a different group. Then, if you are performing comparisons within that group, you can highlight sequence diversity with similar function. So this kind of comparison, so within the group and between different groups, allowed the researchers to say that for Ebola virus genomes, they had very similar but different, sequences in general, but different in terms of intergenic regions and genes of specific functions, and this pointed out to potential vaccine candidates. And so this is highlighting the importance of performing these kind of comparisons. You have here another example where you can be, you see these. This is one unique genome, but you have multi-level comparisons, and here you have the percentage of GC that has been related to specific regions of virulence. Here the same thing in terms of comparison of different strains. So this is the kind of representation that we are using nowadays. This is a circus plot representation, and each line here would show a different genome. So each circle is a different genome. So here is the comparison of all the strains. Here is the comparison particularly of Enterobacter strains. Here the Erwinia strains, here another one, the Panthea strain. And what this kind of comparison can highlight is basically the difference in terms of whatever the feature that you are comparing, like here the GC content again, and this helped the researchers to highlight specific virulence-related regions for the Enterobacter strain, for example. So basically these are a way to represent the different genomes more easily. Phylogenetic relationships could be inferred from all these comparisons, and you have here an example where you could easily identify the gene gain or loss between the different groups of strains in here. So phylogeny is a completely different field that would require a completely different course on its own. I don't have enough time, unfortunately, to cover these now, but I really would hope that you have, a more insight knowledge on this, and I hope that you will be able to understand the full importance of these phylogenetic trees after multiple sequence alignment. So let's see the rest in the next video.